So I'm very excited to host here, generally speakers who come from machine learning and tell us about how they're developing new methods for biomedical science problems, or also the other way around, how biomedical sciences is really motivating foundational developments in machine learning. And so I'm very happy and excited to have Charlotte Bunne here today. So she's a faculty at EPFL in Lausanne in Switzerland, in the School of Computer and Communication Sciences, and also the School of Life Sciences. And before that, she was a postdoc at Genentech and also at Stanford with Aviv Regev and with Jure Leskovic. And before that, she was at ETH Zurich with Andreas Krause, because he was here on sabbatical, and then also with Marco Couturi, and was visiting here at the Broad actually multiple times in Anne Carpenter's lab and collaborating with different groups here at the Broad. Just really exciting science that she'll tell us today about on modeling perturbations and thinking about what types of machine learning models do we need to advance in order to be able to predict the effect of perturbations, often chemical perturbations. She's done a lot of work on optimal transport, now also a lot of more vision models that we will, I'm sure, hear about today, and then bringing it all the way to patients, either for personalized medicine or also for diagnostics. And so I'm very, very excited about her talk today. So Charlotte, it's all yours.
Thanks so much. Thanks so much for hosting me. I'm super excited. I think actually last time I was here was 4 years ago in MIA, so sometime. Yeah. So I'm going to speak about virtual patient labs, AI-driven simulation and diagnostics for precision oncology. So precision oncology is really our kind of main application domain, but many of the algorithms and methods I talk about, they are generally disease agnostic, so you can somehow translate them quite well. But this is a super exciting area with a lot of complex problems that kind of require a new class of algorithms that we are thinking of in the lab. And so with this, I think that's one of the staples that probably most of you here in the room know. That's a classic histopathology staple of cancer diagnostic and prognostics since decades, and really gives us a good understanding of a tissue architectures, tumor architecture, and ultimately very introduction or starting point for a patient on a often long journey through the clinic. And so we also probably, many of you know that the world has become much more colorful. We have really the ability to now often iteratively stain on subcellular resolution tissues and to understand their molecular properties. We can stain immune cell markers, tumor markers, get a better understanding of the overall tumor microenvironment and their interactions with the immune system. And this is by now only one of those modalities that we can collect beyond basically those like standard of care or routine diagnostics. We can also, of course, look at transcriptomics profile, spatially resolved yes or no. But we can also look at genomics, hematology, radiology. So we have these very rich patient profiles that often are actually time resolved. So we have those data that pass sometimes decades. We have patient profile, patients that come to the clinic again after many, many years. And we have those data that span time and modalities. And so we are in a situation where we need to have an understanding of those data because they all provide a complementary view on our patient. They are complementary and they are also incredibly hard to kind of understand and analyze just taking one modality alone. So analyzing a histopathology is one thing. Analyzing a spatial proteomics experiment is even more complex. And a cross-cohort analysis really requires methods that incorporate them to really have them part of the decision or kind of prepare them to be part of the decision process in contexts such as molecular tumor boards. And we have, over the decades, of course, developed a lot of tools and also an initial set of machine learning models to basically deal with this complexity of data, both on the multi-modality angle and the longitudinal angle. But it's still incredibly challenging to really make sense of this huge amount of data that we can collect of a single patient. And so I think it's not a crazy thought that this is a very good playground for AI, and in particular what we think of like AI agents here to really make sense of modalities, because there's just so many things to consider, so many interactions, so many different data points, so many different tools. So what we looked at in a very recent study is how would we build such molecular tumor board agents? And then as a very first question, because that's a kind of like new step in an unknown field, how would we even set up benchmarks to test agents in this context? Because here we have a particular setting that our exciting or kind of initial benchmarks, they really don't capture the complexity of molecular tumor boards. So we started with an initial set of a benchmark that really tests how well those agents have an ability to reason across modalities and to reason across longitudinal profiles. And this is a benchmark that will continuously grow, so that's fairly small, but a starting point. And in particular, what we show is that given the complexity of this data, giving them actually access to more powerful models, what we call those general models, then generalist models, foundation models, is actually going to always improve overall in performance. So here's a very quick example that's a very minimalistic agent. Giving them here access to actually histopathology foundation model always improves in performance independent of the underlying somehow vision language model or language model that is employed. And so how do we think about this? Like we think about some form of like vision language model that is an orchestrator that looks at a particular patient profile, and we have a particular question, we have a prompt, we have an agent that really works as a collaborator with a molecular tumor board. And while we can look at, and here I'm inheriting basically the symbols of Marinka Zitnik's tool universe, while we have access to all those tools that we have so far developed, we actually have this very big question in the room on how do we actually deal and build agents that can access those modalities that are just far more complex than what we've so far are really comfortable dealing with. And this means how do we have agents that really make sense of this high-dimensional complex nature of spatial proteomics, spatial transcriptomics, and this just being two modalities of many, right? These are the two that we are currently focusing on, but then you can make the same thing about radiology and so on and so forth. So what we built in the lab is foundation models that allow us to have a good interaction with an agent and those models. They give us a good starting point, not only to ask very general questions, very general prompts that those data sets might actually answer, but they also give us a good starting point to really in silico test hypotheses. What does it mean? Right? I have a particular patient profile, and this is a starting point for for kind of in silico making decisions that might benefit from some in silico hypothesis testing. What if I would give this patient treatment A? What if I would give this patient treatment B? How would the disease progress over time, right? Can I map from one modality to the other? You can think of these simulators in a very general, very broad term. And these simulators, we are building them in such a way that they allow us, that we have access with this agent to really somehow steer this hypothesis generation and map back at these insights, and then ultimately either like update what we know, right? Refine our hypothesis, make a particular decision, or actually really closing this loop in this lab in the loop framework that we can find an assay that would even provide more insights. So we start from something and then we make suggestions, what other modalities should we collect of a patient? What other experimental readout is important to collect? So that we have a better idea on actually answering this question or running this drug screen. And so I will be talking today about our initial efforts on starting to build those foundation models that also allow cross-cohort analysis, right? And not restricted to single clinical studies. I will talk about how we build those simulators, and we're actually building them now in kind of connection with those foundation models in the first place. And then how do we actually gain insights from those models, and how do we close the loop in? Really somehow steering experimental design and what modalities we collect of a patient. So let's get started with this very first part. The data that we are working with is highly motivated by the data this is collected within the Swiss Personalized Oncology Initiative. This is an initiative already running since several years, also building on past initiatives that have collected multi-modal patient data, including in particular spatial proteomics, but also histopathology. And in the realm of histopathology, we've really seen over the last 3 years very powerful foundation models emerge. And these models are built on this premise that we train models not only on one data set, but on a really, really rich and diverse data set collection. here particularly on really many different cancer indications collected from many different hospitals and environments. And they're trained in such a way that we are looking at particular subparts, patches, or crops of the image, and we learn an internal representation that gives us a really good starting point to now, with very simple linear probing or random forests, go in and annotate cancer type, driver mutations, and so on and so forth. Why are those models very powerful? These are still RGB images, and over the last decade, we have been really understanding how to build those very powerful vision models. So these are vision models that are usually kind of using a vision transformer in the back that are always operating on RGB. Only difference, of course, here we have very, very large images. But the moment we go to spatial proteomics, right, one of the core modalities that is actually collected within the Swiss personalized oncology, particular for beyond standard of care patients, where we want to have a really good starting point for understanding kind of, is this a good recipient for immunotherapy? What is really going on? Which mutations are driving this process and potential therapy outcomes? We are dealing suddenly with a modality that is not anymore standard RGB. In fact, these are incredibly high-dimensional images, so often we have a 40-plex, a 30-plex. We can go up to 150-plex and more. And it's not anymore just always the same markers, but every clinical cohort is going to have a different set of proteins that is actually measured. Because this breast cancer cohort had this particular scientific question, so based on this, we have designed that particular protein panel. And now, if I want to kind of replicate what has been done for foundation models in histopathology, I need to look at many clinical cohorts, and I need to deal with the fact that I have very high dimensional images, and they all have varying protein panels. So how do we build foundation models for this? And then, of course, how do we build multi-modal foundation models across different tissue data? Many of the building blocks that we are using in those models have actually been described in this perspective that was published just 1 year ago, how to build a virtual cell. And this is really this idea of how do we build multi-scale, multi-modal foundation models that allow us to really connect many different experimental technologies into a system that we can much easier query and use as a ground for hypothesis testing. So, very first step here is to build some form of like patient representation from those modalities that are just not anymore easy to query with a simple VLM. And we are building this for now on one of the main technology used within Switzerland's SPO, Swiss Personalized Oncology, which is IMC. We are building a representation of this tissue, so this is why we call this technology or this algorithm virtual tissues, so that we can have many different prediction tasks. It might be clinical diagnostics, this might be annotation tasks such as cell typing, niche predictions, and then we want to build a form of virtual tissue database that we can now go in and really query for a patient that has just a very similar tissue state. And ultimately, we want to build really these agentic systems to support a molecular tumor board that a clinician can query similar patients of other cohorts and just look at overall their treatment history and context. This is exactly what I described. Here we have sourced 15 different clinical cohorts, each consisting of varying number of patients, across different cancer types. So we have here 8 different organ sites, and you can see every clinical study has a different combination of markers. Overall, we are dealing here with roughly 115 protein markers. So now we don't have RGB anymore. These data are really high dimensional, and we need to kind of think of a new way of tokenizing, because our algorithm cannot just always get RGB. It's going to get any type of protein combination, right? And then a lot of them. So we start really by thinking vision transformers from scratch, in a sense that we need to find a new tokenization system to work with multiplex images. And here comes this multi-scale nature. So first we need to also tell the algorithm about the particular identity of a protein. There are many ways of doing this. We have chosen here for now a protein language model that is frozen, through which we don't backpropagate. But this protein language model here, ESM, from broad faculty Alex Reeves, is going to basically tokenize or give us an identity, kind of tokenize the identity of a protein. And then here we have a fusion where we basically tell the model this particular protein is present here with this intensity at a particular location in the tissue. Now we have the tokenization, multi-scale in the sense that it actually feeds in protein information on the molecular scale. But then we have readouts on the cell, niche, and tissue level, because ultimately I care about annotating cell identities because I want to understand those compositions and cell phenotypes. We want to find interesting niches that characterize, for example, an immunotherapy responder or non-responder. And then here, tissue level summary for now, this is basically patient level where we predict cancer type or like certain overall responses based on an entire summary of a tissue. Thinking those vision transformers from scratch also needs to rethink the building blocks, because we have really high dimensional images, so we cannot just use attention as is. We need to think very similar as people have done for video models, how to build basically attention mechanisms for this data. And we want to have an attention between different markers, because of course we want the model to pick up which proteins are co-localized. And we have attention between space, because we want to understand, how spatial regions contribute to a certain decision. You can see here a very simple comparison that had been at the point where we were doing this study, so last year, the kind of like method of choice. This is a model that uses Meta's vision transformers, basically also those that are used in histopathology foundation models, on multiplex images. And you can see here, number of biological dimensions means the number of markers in this data set. The moment we go to 20 markers, it's really, really expensive. The number of parameters grows very, very quickly. And then also the computational cost, it's really, really heavy. So they don't only have the problem that they cannot encode necessarily a variable number of proteins, and also work on proteins that have never been seen, but they also just computationally don't scale. And they can also not deal with the complexity, because you can see here that the more channels I add, the more I know and the more certain I should be in my prediction. But these are models that have always been designed to get RGB, 3 channels, same order, nothing will ever change. So they actually cannot deal and incorporate new kind of like this complexity of the data that we are working with, right? You would actually likely to see at least, at most, like constant performance or performance increase. So these models are general purpose, right? Because we want to ultimately have them answer many different things, even those that we might not think of initially, right? So we design them for now to be general purpose, but we look at them from a particular set of tasks. Very first one is really understanding how well can they now annotate, help us with annotation tasks. So they can do basically cell typing. We can look at retrieval tasks, right? Like looking at basically similar tissues. Here you have basically a query that always just a tissue. Here top 3 hits, and then we can look at the metadata we have. Do they compare? Is the overall tissue composition very similar? And we have the numbers for this in the publication. Ultimately, right, we want to actually have those methods tell us things that classic bioinformatics tools cannot, right? Do they really provide us a good starting point for biological discovery? Do we find biomarkers that we otherwise, or like that we previously did not know, or that are super difficult to craft manually? So I will talk much more about this in the later part of the talk. And then this is something that's not out there, but it actually provides us a really good starting point to now go in and actually simulate in silico drug screens on the tissue. So here we are working with a collaborator that is able to run basically drug screens in tissue. And it's really a good starting point to simulate the drug response of the tissue and how the tissue remodels. And there's a stark contrast to not using foundation model, because that's really, really tricky to think about algorithmically in the first place. They give us a representation that is really good for now generative AI modeling. So just here a very quick example of a setting where we are looking at annotation tasks, but here an even more challenging setup than before. So we are evaluating a model that has been trained with primary lung cancer. And now we are looking at the 0-shot, like basically once this has been trained, what we do is we actually training, so we have a zero-shot model in a sense, or we have a frozen model and do zero-shot evaluation because we are mapping in here a different small lung cancer tissues, like clinical cohort. But it's not only zero-shot in the foundation model training, but we also basically freeze the annotator of the cell types. So here we are testing really, can the model transfer cell labels without any new annotations? We don't need any previous annotations. How well can we actually classify cell types? And here you can see on the left is basically the ground truth. Though ground truth here also is not necessarily ground truth, right? This is just what the annotation has been using classical software. And actually sometimes we believe that these are not very precise. In the middle you can see ours. And on the right, you actually see a method here from Harvard Medical School, Kronos. And you can see that these systems, they're actually not so well working in terms of generalization, because what do you need to achieve here? Suddenly you have a new clinical cohort that has a new set of markers, and you need to really require a model that learns a solid representations that is basically marker agnostic, that you can map in from different protein panels. And this is here, you can see where you actually have a shift in performance that is quite stark. What we right now build, and this is done but not public yet, is actually a multi-modal model. Just want to mention here that this is also a huge effort in terms of like data collection. What we are doing is we are building those systems in such a way that they are open source. We provide the training code, and we also provide all the data that we have collected. So this is publicly available data that has been collected from 1 million different locations in the internet. And so we are also going to release basically the overall data set so that the community can start building and constructing models based on these like somehow multimodal data. This is a model that can be trained multimodal from both H&E, spatial proteomics, perfectly aligned, partially aligned, not aligned at all. And also here, just to acknowledge for the work before and for this work that we just finished, why can we build such models? These models are not like language models, several billions, but they're still 600 or 300 to 600 million. Actually, at this point, there's no point in scaling the parameters further because we are still very, very data limited. But you can see these models are really expensive, and the only reason a lab of my size can build this is because we have been generously funded by the CSCS, 500,000 GPU hours for 1 year for a very small lab like mine. This really lets us think big and bold, and we can build models that also require lots of iterations, testing, and training. So this is the very first step, right? I will show you in the third part of the talk how this really enables us to kind of like close the loop, have novel insights. But before that, I already hinted at this being a starting point for building simulators based on those data. This is work that I showed you that is not public yet, but I will show you a couple of previous works we've done and the same algorithms we are using now to build basically these generative AI-based simulators, and why we even need generative AI in the first place. Usually we have here not cells in tissue. It's much more common to have actually cells in suspension. But what we have of a patient is a measurement of a set of cells. In the first part I showed you, for tissues, we are looking mostly at histopathology and spatial proteomics. Here again, we are actually looking at spatial proteomics data. So we have again a multiplex readout, but this can also be RNA sequencing profiles and many, many other different technologies. And so why do I need generative AI to simulate basically these patient effects? What I want to simulate is in silico, how would a patient respond to a drug, right? How would a patient progress in disease? And what is the most likely state? Because if I can now in silico screen many, many different drugs and overall have the kind of quantify the effectiveness, it will give us a good starting point, right, in particular within the molecular tumor board, to basically make decisions on what's a good treatment and what might be a good path forward. And this is not anything crazy, right? So the molecular tumor board in Switzerland, in patient cases that are basically beyond standard of care, actually routinely runs those drug screens. So we want to be able to kind of forecast a patient that we have measured in terms of we have a subset of the cells that we've collected. And now I want to forecast how they would respond to a drug in a sense that I really forecast or simulate for every cell how they are going to respond. And how they are going to respond in a sense that how do those cells respond to a drug. I want to do this on the level of single cells, because cancer is a heterogeneous disease. We have highly heterogeneous tumor environments. We have very, very different cell types, and it's much more difficult to really perfectly identify subpopulations and then do this based on a subpopulation level. Then we can also right away go on the level of cells. Ultimately, what we are going to extract is, for every cell, we are going to forecast how they've changed over time. And the challenge here is that we need to use generative AI, and we need to model this on the level of a distribution, because every time I take a measurement of a cell, I destroy the cell. So I have unpaired measurements before and after. I collect cells before and after a drug perturbation. These are different sets of cells. I don't have this pairing, so I really need to have a generative algorithm that predicts and forecasts on the level of a distribution over cell states how they would change upon perturbation with a drug, or perturbation with any other kind of perturbation of choice. Then I have a pairing that really allows me to extract how every individual cell has changed. And I want you to remember this kind of change here in terms of like difference in feature expression of all the features that we measure, because you will see that this is really critical to understand the effectiveness of the drug for a patient in the end. There's a lot of different methods that we have developed, and the results I will show you, they still rely on the very, very initial set of methods that we've developed in this space. So here the idea is really to learn a map that parameterizes the change of a perturbation in a one step, like basically one time step. So we really map the cells from an unperturbed to a perturbed state. These are these kind of optimal transport-based methods. We are learning a map. The method of choice here is CELOT. The extension that we are using is CONDOT, is a conditional optimal transport. There's also really nice connections to dynamic systems theory. Back then, this was not a very efficient method, very beautiful, but there are people who have developed JKONet++, works much better. And then there's these really nice connections to control theory and diffusion models. That's the class of models that we are still working with, and there will be new work out in that space. These are really, really powerful algorithms. There is a nice connection in terms of mathematical connection between this optimal transport and this being basically some form of continuous entropy regularized transport. But they are also very, very powerful in the terms of architectures, and we all know them from stable diffusion algorithms. The difference here, of course, is that we don't generate from noise some data distribution, but we really learn this distribution shift from one data distribution to the other. We gave a tutorial in 2023 in the International Conference of Machine Learning to really like kind of connect the dots, how they are mathematically connected, the underlying principles. And we also wrote a reviews methods primer. This is not the only primer, there's also one by Caroline. Everything optimal transport in spatial and single cell omics. So there's many, many application domains for optimal transport that I didn't mention here that are also super exciting. So what we have now is that we are able to predict for a particular treatment for every individual cell how the cellular basically profile changes. And here, just showing you how this kind of like results look like for two different drugs. So here for panobinostat, for crizotinib, in form like for two different patients that he identified with like somehow acronyms or random names. And what we want to learn over all features, and here I show you two, how this control distribution, basically the state before treatment, changes after treatment. So this is here basically distribution for a particular patient, for a particular drug, for a particular feature of over all cells and how they change. You can see here there's just a simple decrease in SOX9 for this patient after panobinostat treatment. Here you can see for crizotinib, it's actually only a subset of the cells that have been responding. So this has been more or less stable, but here these subsets of the cells of a patient have actually responded to the treatment. And I could show you this amount of like number of features times number of patients times number of drugs of histograms. We also quantify them, of course, in overall distances in terms of like RMSD, correlation, distributional metrics. But here, I think that really nicely identifies the underlying distributional shift that we need to learn. Here you can see CELOT. that learns really nicely this decrease, but also is able to understand, on a distributional level that only a subset of the population changes, versus methods that mostly measure an average, right? And these were methods that have been popular by then, right? There's no comparison to flow-based methods here, but there's still many, many methods that actually overall just measure an average, even more recently published one. And you can see then they just really shift on average the overall population, and they don't capture that only a subpart of the population changes. It's also important to acknowledge that not every truth is in those single-cell profiles, right? So here we have number of previous treatments and driver mutations as information given to us, and of course they're going to influence the treatment response. So here in particular, for dabrafenib, right, driver mutation status is incredibly important. You can see that here, for different patients that have a different driver mutation status, there's actually no response versus a response. And you can see that the previous methods are simply not changing this, right? So here you can see that they all predict no shift, even though this patient is actually responding. The method that is basically cellular just with the conditioning conduct is able to really capture that there is in fact a response, and in patients which have different driver mutation status, there's none. Why does it really matter, right? Like, why do we really need to model this on the single cell level? We have, because this is an observational clinical study that has been run within the Tumor Profiler Consortium in Switzerland. We have actually outcome data. We know how much longer they lived, at least to a certain time, and then basically the future stops. So we can basically identify patients that lived really long or didn't live, like basically passed away very, very briefly after the treatment has been given. And so we would like to understand, based on this, can we forecast the progression in terms of acute and slow progression? Now, right, we are able for every cell of this patient to extract for the control population how they would respond to the treatment that they actually received. And we can extract how they responded on the level of the single cells, because for every cell, we basically have how the feature changed upon perturbation with the drug. And interestingly, it's not the metadata that we have of the patient, right? It's not the state of the disease before and neither really the state of the disease after. It's really extracting how the cells changed that gives us the best starting point to reason or like kind of to forecast progression in terms of acute and slow progression. So it's really understanding how the cells have changed, how the patient's cells responded, that give us basically this angle and this anchor point. So just to forecast, right, like why those foundation model embeddings are important and give us an even better angle here with this method we actually used so far always autoencoder-based embeddings or lower dimensional representation for very high dimensional data. But so you can see here that now we basically do the same for tissue remodeling. And we have here a cohort for which we have samples pre-treatment, on treatment and post-treatment for basically immune checkpoint blockade in triple-negative breast cancer. And you can see here a simple representation of those patients where we know responder status, so we can distinguish responders and non-responders. And you can see that without a virtual tissue platform, it's really, really hard to distinguish those or tell apart responders from non-responders. I'm now going to show you, without any kind of generative modeling on any type of analysis, simply the data that we collect of those patients, and that suddenly with this, in this embedding space, representation space of those foundation models, we actually have a basis to really see that there is tissue remodeling or really like a change in these like tumor tissues upon immunotherapy, and that we see the slow response from pre on post in responders, but not in non-responders. So just as a motivation, I already showed you some like initial results on one of those overview slides, that this is really a good starting point to now go in and actually do this forecasting with the same generative AI methods that I've shown you before. So here we have now a setting where we have foundation models that give us a kind of like new space of interaction and kind of angle, many different tasks that we defined that we, in the molecular tumor board agent we already used as basically kind of like an extension of the current toolbox. But can we extract really somewhat a novel insight and then at the very last part really close the loop? So here the same cohort, right? The question is, or the question that we had from the clinicians that we are working with from the University Hospital in Zurich and the University Hospital in Geneva is, can you give us some more concrete biomarkers that are going to be a good somehow basis of like kind of a good foundation to make a decision if this patient is likely to respond to immunotherapy, yes or no? What are the signatures, or can we extract a new form of signatures from those foundation model representations? Here you see the overall representation that the model learns, and you can already see in terms of like how this UMAP looks like, right? But we don't want to just analyze the UMAP, but you can already see here you have the CD4 cells in pink, apoptotic cells in purple. In blue, you have this very like kind of hypertoxic tumor cells. That it learns in terms of environment, not only cell identity, because it clearly clusters, like here immune cells, there are more tumor cells, but it also looks at certain spatial environments. So we have here a subset of B cells that is close to apoptotic cells. We have here a set of CD4 cells that is close to all those apoptotic cells. And so now we went in and we used zero-shot neotrip cohort, again, these 138 patients, where roughly half of them is responders. And we asked, basically, can you identify the smallest set of signatures that gives us a good foundation to now predict with high precision responders from non-responders? And we found in an iterative way, basically 4 signatures, 2 that are corresponding to responder and 2 to non-responders, that give us a high precision in the overall kind of task of predicting response or non-response. You can already see that these are here mostly tumor cells, really like these like super dead, cold parts of the tumor. Here you have these rich, hot environments. So you can already see that this is not going to be like shocking new biology, but something that we kind of suspect already, right? So here you can see the performance increase we have in this response, and this is compared to the original nature method that is basically, it's a great paper. It's all about actually already finding machine learning-based biomarkers, but incredibly handcrafted, that doesn't enable us to actually do this now for a new cohort as well. In gray, you see the clinical stratification methods that the clinicians ask us to run that are kind of commonly based on certain CD4, CD8, or B cell ratios in this tissue. And the reasoning behind this is why we went here is that usually what people do is they are identifying a particular cell of interest, they quantify them, they count them, they are looking a bit of kind of neighborhood studies. How does this translate to a new clinical cohort? Now we can in an automated way, look at those biomarkers, and we can actually look at which cells did the model actually look at. It's looking at particular CD4 cells that are on average much closer to the apoptotic cells, much closer to tumor cells. And this is just like touching, the surface in terms of evaluating really the things that the model is looking at. We need to go much further. Same for B cells. On average, is a particular set of B cell here, and those apoptotic cells it's looking at. Again, B cells that are really in these like somehow active, hot immune environments that are very, very close. And interestingly, there has been a Nature study published 2 weeks ago, so after we finished this, that is a very close description of what we find here in responder cluster 2. But now, right, great, we had zero-shot identified them, but like, can this translate to a kind of new cohort? We got a bit lucky because this has been done in a setting where the foundation model, virtual tissues, has been trained on triple-negative breast cancer in a fairly large cohort. It had now 1 cohort to identify those biomarkers, and then summer 2025, this paper was online, again, a triple-negative breast cohort that we could use as validation clinical cohort. And we find the same set of biomarkers. And in fact, they give us a really good foundation, again, for stratification. And this stratification is better than the original biomarker from the cancer cell study, and again, better than clinical schemes that we use so far. And this is here based on concordance index, so it's a bit hard to interpret. But here we actually don't have responder signal, we only have overall survival times. But you can already see, and in particular when you compare to this one that has really just high risk of only 5 individuals, these are really, like, it's a really, really nice stratification just already checking this visually. So the very first question the clinician asks, okay, so now what's your confidence, right? And this is a lot of work now to really like somehow understand like how confident are we that this translates. And this is only done for triple-negative breast cancer, right? But we believe that the more data we collect, the more we train those models, we can repeat this basically across cancer types. And now what we are doing with a collaborator, really go in and try to get more insights into these particular CD4 cells with additional assays. So how do we do this, right? That basically brings me to the very last part. Ultimately, what I want is that we are able to kind of screen this much more efficiently for patients. Like, what modality do I need to make those type of like, we have multimodal models. Do I always, do I really always need a spatial proteomics experiment? And then I would like to have, for all patients, if necessary, really a spatial proteomics assay run. This not necessarily always needs to be a full 40-plex, right? What are the 5-6 markers that we need to measure that give us anchor points so that I have basically this initialization and this representation space good enough to do those downstream tasks without too much loss in terms of performance. And the motivation here is really that University Hospital in Geneva, they have a Akoya phenocycler codex that can easily do the 40-80-plex, very, very expensive. But inside the pathology, and this is not anymore oncology department, this is worlds apart, they actually have a phenocycler that can do 6-plex, right? And there it's really like a question, which 6 antibodies should we look at? The way we look at this is from an active learning perspective. We have our foundation models, and now we would like to understand what do I need in terms of which modality, which diagnostic test. And for now, we have only 2, but you can think about this much more broadly. To have this representation, to do those prediction tasks. And now we have an RL, or like active learning framework, that sits on top that really decides the policy overall. We call this MAVIS. For now, this is just a spotlight in a workshop at iClear, but we are working on the full version for the January submission. So what it is, it's a multimodal active view selection, we call it. So really at test time, we want to learn a policy that tells us if we need to jump from one modality to the other. Modalities here are sorted in terms of cost, but can also be sorted in terms of availability. And you can put this here into the reward so that it's influencing really when to change. And then we can decide when to stop, early stopping, or if we should explore basically one modality deeper. So here we are sequentially selecting the markers to measure, which doesn't really make sense, right? Because we have either 6, or like we have them basically in batches of 5 or 6. But I mean, still we do this now for simplicity. We have a reward that for now takes into account the kind of downstream type of interest, but now we developed it in such a way that the model from an internal state in a self-supervised way can actually optimize the policy at test time. And now you can see here just for this subset of just designing the assay. Here this is for data where we don't have multimodal data. We not always have associated histopathology. We ask an expert, meaning basically the person in SUG that actually runs this experiment, to design us sequentially a panel, which doesn't make a lot of sense. Like usually they have their like kind of panels and they can roughly sort them in importance, but it's not really like a perfectly sorted list. But you can see that the first 3 are the obvious ones, to map out the nuclei, to map out the tumor cells. But then afterwards, it's not so obvious anymore which signal the model needs to really embed already at a level of 5 markers. Here select as few as number of protein markers. What is really giving the leap in performance here almost like by 10%. We can compare also, of course, with the protein language model. Can I just kind of like sample diverse proteins? Here, this is these ESM baselines. And here's a measure of uncertainty that's actually basically like kind of in terms of likelihood defined. And it slowly goes down the more views we take. Now we can go one more extreme step, right? Like, can we do this for not only for experimental design, but also for modality selection? And we know that for certain tasks, having this like 30-plex spatial proteomics here actually gives us quite a boost in performance over overall H&E in particular, because we have here a lot of TMAs where the histopathology foundation models struggle a bit. And you can see that we want to reach the same performance as always running the spatial proteomics full plex, even though we actually, in most cases, only ask for the H&E, and then never more than 6 markers. And you can see, and this is log scale, this is really cheap, $2,000 very expensive. And then on average, right, we would actually spend $50 per patient. So this becomes feasible, also becomes feasible because we realize, I don't have this here, but not every patient has a randomly different set of markers, but we actually see clusters, certain patients requiring different assays. So there's clear groups in the patient profiles. And yeah, so with this, kind of thinking this more broadly, right? So we are working very, very closely with this molecular tumor boards. That's just like some crazy fantasy. We have a really, really nice infrastructure in Switzerland in terms of Swiss Personalized Oncology initiative that I'm highly benefiting from, right? Like starting my faculty position last year. But now we've really recently started something that we call a kind of national AI initiative in precision oncology. It's a 90 million Inno-Swiss flagship that you can see has 4 out of 5 university hospitals in Switzerland, has regional hospitals and private clinics, has all the important academic partners. It's led by EPFL. And really thinking of, okay, how do we build our infrastructure that allows us to connect, our supercomputers and Swiss Data Science Center to places where the data is collected? Because now, I slowly manage to get all the data out of the hospital environments in terms of tissue data, but genomics data is a whole different beast, right? There we really need to think about secure but AI-ready infrastructures, to think about AI-powered molecular tumor boards, which is the centerpiece of this grant. And yeah, with this, thanks so much. So most of the work has been done by my students. So there's Johan, Ishan, Benedikt, Lukas, Yexian, actually also Alexandre, the very first work. There's a lot of work that I have still mentally done with my PhD advisors, Andreas Krause and Marco Cotturi. Had a lot of ideas here at the Broad, in particular in terms of imaging data from Anne Carpenter and Shantanu Singh. And then I had a really productive, kind of fun postdoc last year with Aviv and Jure, Genentech and Stanford, and really, really driven by these close collaborations with Andreas and Olivier, as well as Lukas Pelkmans at the University of Zurich. And thanks so much.